So it is my distinct pleasure to introduce both of our speakers today. So I'll start off just sequentially. So first, Fabrício Vieira got his Bachelor of Science and Master's of Science at Sao Paulo State University in Brazil. He also went on to get his PhD also at the same university. But with that came a one-year event. He had to come here to Penn State to work for a year and worked with John Pecchia on mushroom production with that. So Fabrício has actually had 15 years of mushroom cultivation experience. So he has quite a bit of experience in this field. And one other thing just to note for Fabrício, and some of you may recognize, he's been involved in the Microbiome Center for some time. And in fact, he gave the very first talk when it was still the Microbiome Initiative in the fall of 2016, before it was officially established as a center. So very great for that. And Eoin O'Connor did his bachelor's and his PhD at Maynooth University in Ireland, also where he gained a lot of experience working with mushrooms in other fields. He joined our labs here in 2022 at Penn State. And two of the reasons, in addition to his experience with mushroom cultivation, is his experience with proteomics. And that was one of the reasons why we really recruited him. So one last thing just to wrap up to say, the work that they'll be presenting is funded. It's a collaborative project between my lab, Carolee Bull's lab, and John Pecchia's lab that was funded by a grant from the Huck Innovative and Transformational Seed program in 2020. And so we're very thankful to Huck for giving us the money to get this project off the ground. And I think you'll see with the work they're going to present that we've really run with this project and are very happy with that. So with that, I'll turn it over.
Morning, everyone. Thank you for the introduction, Kevin. Basically, you said that I'm a fun guy, right? So today we'll be covering some partial and final results of the HITS funding project, Unlocking Mushroom Devomes, Pioneering Mushroom Development Research with HITS. My name is Fabrício. I would like to start with those illustrations. Between those two photos have more or less 350 years apart. So we move from underground to above ground cultivating mushrooms. That picture or illustration in the left, on my right, is from France, when everything started regarding white button cultivation underneath Paris, and then they move above ground over the years. In terms of microbiome, how much we know about mushroom microbiome? Just a quick dirt search using Google, you can see that human microbiome have much more results, which makes sense, right? We should be in good shape and health to then go cultivate a mushroom. Environmental microbiome, right below, agricultural microbiome is gaining attention, and then is us down there. The mushroom microbiome. But impressive is a big number too. Of course, those results was a quick dirt search. It's not using Web of Knowledge or some scientific database, but I think illustrate the differences in knowledge using different systems or kingdoms, whatever. Specifically about white button mushroom cultivation system, how much we know in the past 100 years. So at the beginning, I usually say in a classroom that we are cultivating microbes in a large scale. I don't work with plants necessarily, not animals, but I grow fungi. So it's a lab in a large scale. We have the concept of lab in a farm when we go grow mushrooms. So sanitation, sterilization, pasteurization, all those concepts apply in microbiology we use as a farmers. What the hell is going on? So at the beginning, or 100 years ago, more or less was how to make those compost piles heat up fast and provide a better food for the mushrooms. So that was the principle of microbial ecology, which makes sense, right? You manipulated your pile of compost by engineering a process that will help the microbes degrade that material fast. So later on, we start identifying those organisms, the major players. Then later on, we thought, oh, okay, we can reintroduce those microbes and speed up the process. And then we are changing our view of microbial communities. So we are looking as a network, which makes things a little more complex. Penn State have a huge importance in the white button cultivation systems. One of the most successful patents for Penn State come from the mushroom group with the spawn. And also in the 20s and the 40s, Penn State helped define the modern composting process for white button. So Penn State is a powerhouse in terms of mushroom cultivation knowledge. The research program is around 100 years old. I just want to sell and highlight the Penn State importance for the field globally. Talking specifically about white button cultivation system, I call a microbial community flow. Why? Because we take different materials from different crops, which have different microbiomes. Cornfield have a different microbiome that are chicken guts. Why chicken gut? We use chicken manure, horse manure. Sorry, I don't know what's going on here, but
Must be on the timer.
yeah, right. So we have all those microbiomes from different environments. We put them together in a compost pile, feed them, and make them degrade that material, which later will be selective for white button. So the process is very short. In 12 days, we have raw material and we make soft, decompose that material. We artificially speed up the decomposition process that naturally happens in the nature. So after that, we inoculated right here at the end of the composting with the white button inoculum. We have compost colonization. After the compost is colonized by a white button, we add a topsoil layer, which is typically peat moss. The fungi will keep growing, colonize the casing layer, the peat moss. When they colonize halfway to 3/3 of the casing layer, we start inducing fructification. How? Changing the room, the environmental control in the room. Temperature, CO2, O2, and humidity. So then the mushroom shows up. We have 3 flushes, or 3 harvests, or 3 breaks, 7 to 10 days each. So the whole process happened very fast. 50, 55 days, 60 days. We have raw materials. We make the fungus food. We inoculated the fungi. We manipulated the vegetative growth, the reproduction stage, and then we harvest the mushroom, cook out that compost, that material, which can be reintroduced in other ag crop system as a fertilizer. It's an interesting system to work with because a lot of things are going on here. We have thermophile phase, mesophile phase, and even cold phase. So sorry about that. For those that have interest in working with microbiological systems, we have everything in one here. We have hot, cold stages, shifts in community, different microenvironments like compost, peat moss. Diving in the project itself, the Devomes concept was initiated by Dr. Monica Medina when 20+ PIs at Penn State and outside Penn State got together to apply a grant to NSF, Science, Technology, and Centers. Devomes means developmental microbiome, and with Carolee, Kevin Hockett, John Peck, and my boss, Owen, and I, we put this to work. The HEATS project, demonstrating and manipulating mushroom Devomes, have 2 components. One conceptualized by Carolee Bull, the Subtractive, and one conceptualized by Dr. Kevin Hockett, passaging. The first one, subtractive, is a chemical approach I classify as a chemical intervention, and the second one, passage, as like a natural re-inoculation of mature communities. I will explain during the presentation what that means. So the first one is a subtractive component. It's a little busy there, but I want to divide or simplify in two portions, two components, in vitro and crop trials. We make a screen or a selection of a diverse array of antibiotics that people tested in close related ecological niches, which is soil, basically. We test them against agaricus only in vitro, petri dishes, then in microcosmos using compost, and then after that, we went to crop trials. Starting with the component one, so we have 43 antibiotics that belong to 10 different classes. We extract them from previous work on literature, and then start the challenge. Our organism is a slow growth organism. So that's a representation there, like 21 days to colonize a specific designed media, compost media, malt, PDA, all the medias we use routinely in a lab doesn't work well. So how I can measure antibiotic effect if they don't grow fast, right? So, and I also have random growth. This was not induced, but it's cute, right? I mean, they're growing. Just to demonstrate how the behavior of this guy in petri dishes. So we come up with like a easy kindish assay, 5 millimeters from the plug, mycelium plug to the filter, like tiny. I have 4 days to measure the growth. Essentially, out of 43 antibiotics, I know you guys maybe have interest in read all, but you don't have time. But we can talk later if you guys have interest in know why we choose those specific 43 antibiotics. But just 1 out of 43 have a slight impact in the mycelium growth in petri dishes. So after that, we make another round of selection to proceed to microcosmos and trials. Make a crop trials with 43 antibiotics is like, we need to double up our facility. We don't have physical space to do that anyway. So, but we selected 10 antibiotics from 10 different classes, including with that have a slightly negative impact on petri dishes, to see if that effect goes to the compost or casing microenvironment when are in crop trials. The racing tubes or microcosmos, we inoculated, we spread antibiotics in compost and inoculated with agaricus and measured the mycelium growth. So the impact was kind of similar to what we saw in vitro. Just one antibiotic have a little effect in mycelium growth. Okay, we decide, well, by design, right? Antibiotics does not have too much impact in fungus or fungi in nature, right? So we are happy with that result, kind of a confirmation. We proceeded for crop trials. Well, sorry. We did a microbiome screening on those racing tubes with compost and antibiotics. So 10 antibiotics plus solvents, and we have compost without agaricus and compost with agaricus to see if the agaricus itself are reshaping the community. Looking at high taxonomical level, not much shift, right? Similarities in community with or without agaricus. But then when you start break down to diversity, alpha, beta composition, you start to see some interesting things. In the presence of agaricus, the community shifts in alpha diversity. Without agaricus, just compost with the natural microbe that was there, plus antibiotic, not too much change compared with the standard, right? So that was kind of interesting, and we have publications before that kind of show that the host, or the agaricus in this case, reshape the compost community after inoculation, as well reshape the casing community as after inoculation. The composition was similar in my right panel here, or your left. We have presence and absence all together. We can see a separation between presence and absence. And then when we break down just for presence at the end of spawn run, which is the colonization period, 16, 18 days, we saw some shifts in community. Look into who is more or over or underrepresented. So because rifamycin have some differences in vitro and in compost, we decided to make a comparison here. We found some genera, but we don't have too much information on literature of the functionality of those bacterial genera in our system. So we have some work to do. As usual, you start with some questions, you get some answers, but you have more questions after, right? Most of the time this happens. That's the end of the in vitro component. So we kind of confirmed that antibiotics does not have a direct impact on agaricus mycelial growth, but looks like have an impact in the community. So moving forward to the crop trials, or the second component, we applied first antibiotics in the compost microenvironment and then in casing microenvironment, two separate trials. The effect on yield in mushroom experiment. So how we count this? Total yield and by harvest. So break 1, break 2, break 3. So 3 weeks of harvesting mushroom. They comes in waves. 3 harvests, 3 breaks, 3 flushes. Looking to the total yield, we can see have some variability compared with standard. Most of the statistical differences here is between the lower yield treatments against standard or untreated compost. We have some variation here with water because we added 1.5% of water to the compost when we apply the antibiotic solution. So we kind of expected to see some variation even for the solvents only, in this case water. But also work with mushrooms is a little different than plant and animal. We have randomness there. So we have that mycelium network that we don't know how many mushrooms will come out if you are not managing the room properly. So we are kind of happy with 10% variation in terms of yield for any mushroom experiment due to the nature of that system. Moving forward to community, what happened with the community? So I organized it by solvents and antibiotics that use those solvents. Again, high taxonomical level, not too much differences. But when you start breaking down, you start seeing interesting things. Most of treatments showed a decrease in alpha diversity comparing with untreated compost, right? Always, I try to keep the same standard, the first one, and then the treatments. Treatments with a decrease in alpha diversity tends to cluster close to each other in terms of beta diversity. So decrease alpha diversity, you kind of get them more specifically in the same group, even if you are using different antibiotics. That was for compost. Talking about casing, we saw more dramatic shifts in mushroom yield in casing compared with compost. You can see here a big drop for this group of treatments. How we measure mushroom yield? Kilos of a mushroom per square meter of a mushroom bed. right? There's different units or measurements comparing plants and animals. So for us, is area, kilos of mushroom per area. How about the community? We saw some shifts in high taxonomical level, differently than compost, start like in a high hierarchical taxonomical level. We saw similar trends. Most of the treatments decrease alpha diversity and similar to better composition. So those treatments have a tendency to be close to each other, right? It makes sense. You are decreasing the alpha diversity. This component was the heaviest one to manage because it was 18 months of funding and you need to show the results. We need to do crops, set up everything. So it was a little challenge for me, but we did it. And we have some things to validate. Summary, looking far away, like I said, I'm still working on this part of the project. The compost community seems to be more resilient in terms of dealing with biocides, in this case, antibiotic, which I was expecting that as a fungal ecologist. So they're more resilient and break down things that we don't want there. Increase in alpha diversity, not necessarily increase mushroom yield. Shifts in casing communities appear to impact mushroom yield because the cases where the trigger happened between mycelium to fruiting body. So that portion of the system is very important to make fruiting bodies. Most of the bacteria generally shifted in compost and casing communities has no function in the system. Like I said, I have more questions now than before, but I think that's good. We are moving forward. So that was the subtractive component. Moving to the passaging component, which is two parts. The descriptive part, screening the community, and then comes Dr. Eoin O'Connor talk about potential functionality of those communities. The idea here was to reintroduce mature communities at the beginning of the cycle. So mature communities from compost to the beginning of the compost or to the beginning of the colonization, and mature community in casing to fresh casing raw materials. That happens around day 10 or 11, where the mycelium start changing, making nodes and forming pins, baby mushrooms. So we took the samples right there when the mushroom was changing the life stage, and we reintroduced to respectively microenvironment, compost to compost, casing to casing. So what happened in terms of mushroom yield? We did not see any statistical difference in mushroom amount, but we saw change in frequency. So when you re-inoculated a mature community from casing to fresh casing, we saw anticipation in mushroom formation. 4.5 days early, we start picking mushrooms early in that treatment. So that's very interesting for a perspective of automation or how the industry is looking to have a more vertical cropping system. In the future, you can spread the harvest through the cropping period. That was interesting for applied perspective. How about the community? We saw that compost and casing are two different entities, right? Compost, basically, for us here is the wheat straw, horse manure, chicken manure, gypsum. Casing, peat moss, sphagnum peat moss, wetlands, acidic, high in water content, two different ecological niches, right? And we mix them together, boom, we have mushrooms. But we confirmed that they have different community profile, different attributes, microbiologically speaking. And on top is compost, and the bottom is casing. We noticed that no difference was observed between standard and passage compost or enriched compost, but we saw differences in casing, which is correlated with maybe the anticipation in mushroom formation. The trends during the crop was kind of similar. We have a tendency to compost community decrease, and we have a tendency to casing community to increase in diversity over time during the crop. But similar trends, no statistical difference between time points in compost or time points in casing during the crop. About beta diversity or composition, like alpha diversity, passage casing showed significant differences in community composition. You can see here that's more clear. It's clear to see the separation here in casing comparing with compost. Sorry about the statistics, the asterisks. So because it's kind of a large number of treatments, you guys need to trust me. Or maybe read the paper when you guys have interest. We did like over and underrepresented genera, and we found some, but most of them, we don't know the function in the system. We are working on that to try maybe do some in vitro trials first and see if we can capture a larger picture in the future. Summary of passaging. Passages of microbial communities appear to speed up compost and casing colonization, but did not produce more mushroom necessarily. Passage of microbial communities display a potential to encasing to maybe change the mushroom frequency in a growing room. The results outlined here show that earlier fruiting body formation is visible with passage in casing, and it may be related to the transfer of bacterial communities in which were identified and can be explored in the future. Like I said, we don't know yet the function of those community, of those individual groups, but we are working on, and that was very important to have such initiative for a high risk research. Maybe we'll lay the ground for future research regarding microbiomes and mushroom cultivation systems. So I will pass the stage now to Dr. Vieira and O'Connor. He will be now putting the berry or the cherry on top of the cake with the potential functionality of what we screened during the subtractive and passaging component.
Thank you. I've never done a dual presentation before, so I guess I should introduce myself, but yeah, I've been introduced already. My name is Eoin O'Connor, and previously, during my PhD, I basically did work on genomics, transcriptomics, proteomics, and joining the Devomes group, I wanted to take some of my experience, particularly with proteomics, and try to apply that at the microbiome level, so I gave myself a difficult task. My work is involved with the additive approach of the passaging, and Dr. Vieira described it in great detail, so I won't go over it too much. But my work, it was related to the use of standard and the passage casing. And as already mentioned, the passage casing is created by taking casing at the point of pinning. So we choose, we have multiple reps, we choose the best performing tubs. We take that casing and we add it to a standard casing. And this creates our passage casing. For the work I'm going to present, this was done with 10 cycles as opposed to 3 as previously described in some of the work. And the idea of this was, again, moving into the idea of manipulating microbiomes. So one theory I have with the manipulation of microbiomes is, do we go from something like a species-rich microbiome and then maybe move to a more specific microbe-enriched microbiome? And within that specifically enriched microbiome, do we see a functional redundancy previously, and then we move into more of a functional enrichment? So moving into this idea of the functional microbiome. So when we repeated some of this work, we were happy to see that we got some of the same phenotypes previously seen. So we had a pinning simulation. So you can see here the CP being the passage casing versus the standard CS casing. We see that the mushroom yield begins at an earlier point for the passage casing. This happens at about an average of 4 days. And I think these images really drive home that idea. These images were taken on the same day of that crop. And you can clearly see that within the passage casing, we see a lot of pins and mushrooms forming and nothing at this point for the standard casing. So pinning stimulation is quite obvious in this regard. As well as pinning stimulation, we want to investigate the potential for passage casing's impact on disease dynamics. And we took a commercially detrimental disease bacterial blotch caused by mainly Pseudomonas thalassei and some other bacteria as well. And within this, we added the cell suspensions to a casing. So in this experiment, we added to the standard and to the passage casing. And then that casing was added to the plots, and then the crop carried on as usual. And you can see from the non-symptomatic yield, which are mushrooms that do not appear to have any disease, we can already see that just for healthy or non-symptomatic mushrooms alone, the BP or the blotch inoculated passage casing has a much higher yield than in the BS blotch inoculated standard casing. We also looked at the, disease severity for a blotch, and we got some interesting results when we actually break down the disease into different grades. So the different grades signify different levels of blotch. Grade 1 being a low amount of disease expression, grade 2 being a moderate amount, and then grade 3 being a severe amount of disease expression. And within that, you can see that particularly if we take grade 3 disease mushrooms, there is a much higher proportion of mushrooms within the standard casing that have this level of disease. And it's much lower within the passage casing. And this chart just breaks down a by day cumulative yield of those mushrooms. So the yield of these mushrooms relates to disease mushrooms in this case and not the non-symptomatic. If we look at the disease progression using AUDPC values, so these are transformed yields into area under disease progress curves, you can see at different days between the BP and the BS and that we have significant differences in blotch yield, blotch infected mushroom yield. And overall, if we take these two treatments into account over the whole course of the crop, there is statistically significant differences in the amount of grade 3 blotch infected mushrooms we see with these two different treatments. So yeah, we can see that there is clearly a suppressive effect when we use passage casing in terms of blotch. We also investigated green mold, which as the name suggests, it's a fungi-related pathogen. It's a fungal disease caused by, in this instance, Trichoderma aggressivum, which is the main pathogen found within the US for green mold. And we saw the opposite effect when we used passage casing. So passage casing actually had a conducive effect for green mold disease. We can see here with the TS and the TP that the yield, and again, we're going back to the non-symptomatic or healthy mushroom yield in this case, the yield of mushrooms from these plots was higher than in the passage casing. This was actually evident within the growing rooms. We could see that there was this effect where with the standard casing where we added the cell suspensions illustrated here of the Trichoderma spores, you couldn't see any green mold on the surface for the most part within the standard casing. However, there was this clear zone of no growth that we could see within the passage casing experimental units. And I believe this was found in 7 out of 9 reps that we had this interesting kind of phenotype within these plots. So there was a very aggressive growth of Trichoderma within the passage casing versus the standard casing. We still did see disease within this, but much more so within the passage casing treatments. So the actual treatment itself created this conducive effect for this disease. Looking at the overall yield within the crop, the CS and CP are the control treatments, so standard and passage, Trichoderma and the blotch here. Within the total, within the first harvest, second harvest, and third harvest, looking at blotch, there are significant differences in yield between each. For the Trichoderma, we could see that there was a statistical difference within the first and third harvest and not in the second. But we do expect to see the significant difference within the first flush because if you remember, passage casing does stimulate pinning. And this was even evident when we had the pathogens introduced, be it green mold or blotch. So that would have driven this difference here, particularly with the Trichoderma treatments. But this just shows us overall that there are much more differences to be found with blotch that did that for me. So moving on to some of the functional work I did. So moving back to just the idea of the differences between standard and passage casing, removing the disease element for now. I want to introduce the concept of metaproteomics, and anyone who may have heard me speak previously in these seminar series will know that this is what I like to harp on about a lot. But metaproteomics is the use of proteomics at the microbiome scale. The term was first coined in 2004, or at least first appeared in a publication. So it is still in its infancy. It's only 20 years now since that point. And I produced this graph just to show that how quickly the exponential rate of publications have been over the years when we look at the term metaproteomics within PubMed. And I predicted that by the end of the year, there will be over 1,200 publications using this method to analyze the functional microbiome. Most of these publications do exist within the medical sphere, wastewater treatment, but within the horticultural sector, it's not being adopted as quickly as some other fields. So we're hoping to make some changes in that regard. With metaproteomics, some of the benefits include a much better idea of the functional processes within the microbiome. So not only can we see the number of different microorganisms, we can also get an idea of the metabolic processes that are really enriched within certain treatments. With metagenomics, we still get the best resolution of taxonomy, but we can only really have a predictive level of function within this technique. Metatranscriptomics does have a higher level of functional annotation that we can derive, but really metaproteomics shines when it comes to the idea of functionally what is happening between microorganisms within a microenvironment. So it's a new technique, and with any new technique, there are always challenges to overcome. And some of the biggest challenges relate to the databases we use and protein inference. So starting with database, there are 4 main different categories of database that you can use for your metaproteomics. So with any proteomics experiment, we generally have maybe a single genome. We get a map of potential peptides from that genome. And then what we actually observe from the mass spec is then mapped to that in silico map. And then we can pull out what proteins are actually present based on the genome that we have available. With metaproteomics, as you can imagine, you have hundreds, sometimes thousands of genomes. And that's where a lot of the issues, where we find a lot of the issues with this technique. So one of the best ways to overcome this is to use a matched metagenome. So as the name suggests, you take DNA from the same samples you ran from your, you extracted proteins and ran on the mass spec. And with that DNA, you can do shotgun metagenomics and create your own database based on MAGs or metagenome assembled genomes. There's also the unmatched metagenomes where you can simply download genomes that you predict to be within your microbiome, but this takes some a priori knowledge of what might be in your samples. There's then the unrestricted reference database, which is just basically looking at all proteomes available on a large database, such as like UniProt. And then the restricted reference database is a restricted version of number 3, where we can download certain, or we can infer certain proteomes based on what is publicly available. I have these 2 highlighted because these are 2 different techniques I used. And within the protein inference space, with any typical proteomics experiment, we generally apply Occam's razor, where the easiest path to the answer is generally the correct one. And within our raw data from the mass spec, we have PSMs, or peptide spectrum matches. And generally, peptide spectrum matches are assigned to proteins that are predicted from the genome. And the least number of PSMs that are assigned to a protein is considered a positive hit for that. And there are always false discovery rates and different algorithms applied to actually that matching. But with metaproteomics, as you can imagine, you have a microbiome, you're going to have similar strains, you're going to have similar species. So Occam's razor is actually counterintuitive to those peptide spectrum matches. And within that, we do something called we use a technique called the metaprotein, where instead of saying this protein is assigned to this organism, you might have a metaprotein with a broader function, or a protein that is assigned to a larger group of organisms based on lowest common ancestor. So within the restricted reference database, I did have a few different analyses that I applied, but I just want to point out one particular result from using this kind of database. I used a program called Umpire, which uses parsimony, where you can apply, or you can provide it with mass spec raw files. you can give it a list of genomes and it'll infer proteomes based on the genomes available, based on the mass, the raw mass spec files. Umpire has all bacterial genomes within it, and then you can apply your own genomes. So in this case, I added these fungal genomes. And from this, the system predicted 8.3 million sequence FASTA file based on what I provided it with. This is a very large database, and it creates a lot of downstream parsing challenges. And within that, then I use a program called Peaks from the proteomics core. And Peaks is quite beneficial for metaproteomics because unlike other search engines or systems that have search engines, Peaks actually does de novo peptide predictions. It'll bin de novo peptide predictions and then use those bins to actually run it against a database that you provide it with. So it's more of a two-step kind of search system. And then this is just one thing I want to point out with the restricted reference database. So these are chord plots where we can look at the functions on the left and the taxa that they apply to on the right. And within the restricted reference database, I do have the benefit of being able to look at agaricus proteins in tandem with some of the bacterial proteins of interest. And I just pulled out this particular results just to show that within the gamma proteobacteria, which contain the pseudomonads that we're always interested in, you can see different terms relating to transcription, defense. Particularly receptor proteins are always an interesting result to see because we're always interested on the interface between the host and these potentially beneficial bacteria. And you can see that this is signifying a high growth rate, metabolic rate, and I guess a happy situation with some level of competition with other bacteria. But what I really want to point out to is this term is the most enriched within these systems, so the highest in abundance is the monoatomic ion transport, and this appeared within the standard and passage casing. Here I have day 5 just for as an example. But you can see here that these are being assigned to bacteria, so it's not really informative as to where it's coming from. And this is one of the issues with using such a large database. So this would come down to, yes, we're getting some valuable insights, but we do still have the difficulty of protein inference, particularly in terms of lowest common ancestor. It's at a very high level here with bacteria. So some of these results made me change my approach and look at the matched metagenome method. So just to mention the kind of proteomics I do is shotgun proteomics. I do LFQ quantitative proteomics where I don't apply any tagging to my proteins. I just basically extract all the possible proteins within the system. And this is an illustration of how that is done. I don't have time to really go into too much detail, so please ask me afterwards if you're interested in knowing more. From these samples, then I took day 5, 10, first, second, and third flush, and these casing samples would have then been subjected to protein extractions and then subsequent mass spec. From these same samples, I took DNA and performed the matched metagenomics. So this is a quick illustration of how I created my mags. In particular, the one important step was to remove host DNA from these genomes. And then these would have been assembled into one large database. This database contains 69,000 sequences specific to my samples, which is obviously a lot more manageable and specific based or compared to the over 8 million in the previous method. So from metaproteomics, we can infer relative abundance, and this is peptide-based relative abundance. You could do this by counting peptides within a system, but actually I use the LFQ intensities to actually infer. So how much a peptide was present within the system and how much intensity you had within that spectra was used to infer how much biomass was being contributed by different phyla. And this does correspond with a lot of the results we see from amplicon sequencing. In particular, we see that Pseudomonas data really dominates here. And a quick kind of housekeeping note, I do use the new terms. So Pseudomonas data relates to Firmicutes, and the bacteria, or the Bacillota, I said that the wrong way around. Bacillota relates to the Firmicutes, and the Pseudomonas data relates to the proteobacteria. So we do see a lot of overlap with the amplicon sequencing and some interesting insights. But overall, there's not a stark difference if you look at, so each pair is a standard versus a passage that same day. But there is some interesting results if we look at the point of pinning and even at the first flush, that there are differences in relative abundances of some of these phyla. looking at some of the principal component analyses, the samples group a lot more, the groupings are a lot more disparate when we look at day 0 and even day 5. And as we move towards the flushes, we start to see that these samples start to appear more similar. Using protein expression, we can see that there are clusters between these samples, and where we have more green, there's more similarity. So as expected at day 0, there is more dissimilarity between the standard and passage casing, and then that starts to become more and more similar as we move along the crop. So it's really these two clusters that will probably be a point of interest moving forward to look at what might be stimulating, or what might be triggering the pinning stimulation phenotype that we see. So some of these results show me that maybe pairwise comparisons are going to be more biologically informative for these systems. So again, with the chord plots, because I think they're very pretty, but on the left we have biological processes, and on the right we have taxa. And I actually removed some of the rare sequences from these plots, but even you can still see that they're a bit of a minefield of different functions and the taxa. Here we're just looking at passage versus standard casing. So on the left we have standard, and on the right we have passage. And just from a quick overview of these results, again, we're getting the same terms are reappearing. So monoatomic ion transport is really enriched within these systems. Within proteomics, we tend to use volcano plots. Volcano plots can show us what is statistically significantly enriched for different proteins. So we say SSDA, so that would be statistically significantly differentially abundant proteins. So SSDA proteins give us an idea of when a protein is shared between two different treatments, is its regulation downregulated or upregulated between two different samples. So where we have the red points here, that signifies that it is enriched within this system, and the green would be enriched within this treatment here. One interesting result that came from these, so from this pairwise comparison alone, we have over 3,000 proteins. You tend to, it's tricky to assign SSDAs when it comes to metaproteomics, because the actual stats that are applied filter out a lot of results. So these all go beyond a fold change that we'd be interested in, but I'm only talking about ones that are known to be statistically significant. One thing I noted from these data sets is we have a lot of the term ton-B being represented. So of 3,000, there's over 200 ton-B proteins present, and there's a fold, the fold change of these is 275, and then SSDA, there are 11 downregulated and 12 upregulated when it comes to passage casing. And some of these taxa below just show where these are appearing from. This is not a result that I had my eyes on prior to seeing some of these data sets, but it's certainly something that's jumping out and being very representative. So actually looking at the top 10, so for standard casing, some of the most upregulated terms relate to a lot of nutrient uptake from different phyla. We see some small molecule exchange and signaling. We also see some terms that relate to a sessile state, so immobility of certain bacteria. But really nutrient uptake seems to be what's dominant. And this could actually be a sign that there's a competition between these different bacteria within this system. But then for passage casing, some similar terms are reappearing, but it's slightly different. So the ABC transporter seems to be a really important process within this. We'd see that relates to bacilli. And again, that relates to nutrient uptake. It's a very broad protein, so it could be inorganic ions, sugars, amino acids, peptides, or vitamins. And then as well, we see some stress response. So this is definitely terms that we tend to see when a certain bacteria is competing with another microorganism. And if this is so highly enriched, it could be a sign that it is either doing very well in that competition or it is having a hard time. But we still see a lot of proteins related to signaling, and we see some proteins related to nutrient uptake again. So there's a lot of different processes going on here. One interesting finding is the metalloenzyme. Ton-B is a receptor that's related to iron chelation. And there are some enzymes that are highly enriched that are enzymes that have iron on the outsides of the protein. And iron is required to actually make this enzyme. And this enzyme is related to nitrogen cycling. So then moving into some of the pathways, if we look at COG terms, I wanted to dig a little deeper into the idea of looking at different phyla separately within the standard and passage casing at the point of pinning. And you can actually see within the alpha proteobacteria, there's actually quite a large diversity of different functional pathways compared to the passage casing. These relate to membrane biogenesis, which could tie into the idea of receptors being produced, but it is quite a broad term. And again, we see the iron transport, which is related to the monoatomic ion transport. And interesting here, this points that it is an inorganic ion transport. And some of these terms are similar between two systems. The gamma proteobacteria, we see a slightly different platform. We're always interested in the gamma proteobacteria. It does contain the pseudomonads that we know play an important role in fructification. We can see that within the standard casing, there is still membrane biogenesis, some unknown functions. But if you move over to the passage casing, there is really a much larger diversity of enriched terms within this system for anything from ion transport, vesicle transport, energy production. There's a lot of different processes related to high metabolic activity within the gamma proteobacteria. And then finally, I just want to point out the Myxococcii. So I noted within the relative abundance charts that from standard passage pinning, one thing you can see that there is a higher level of the Myxococcota within the system. It's not a striking difference, but it's a difference all the same. And this difference was highest for this particular biome at this point. And if we look at some of the cog pathways that they're involved in, again, we see membrane biogenesis, we see inorganic ion transport. So these are all pathways that we're seeing reappearing in our systems, and it's creating targets for bacteria that might be important within the casing and what they may be doing. So I think I'm fine. Yeah, so just to conclude, obviously we presented a lot of work here today. So this is mainly a conclusion of what I showed you. So passage casing stimulates primordial formation and suppression of bacterial blotch in mushrooms. From the SSDA or fold change values, we can see that some of the proteins that are dominant relate to nutrient uptake and a potential competition between different bacteria is seen within the standard casing. There's a difference within relative abundance between standard and passage casing, and these are most notable within the Pseudomonadata, bacteria data, and the Actinomycetota. We could see within the functional microbiome that there's terms related to metabolism, membrane biogenesis, and vesicle transport. And just to really wrap up, our understanding of the mushroom devome is still in its infancy. This project has pioneered research into the influence of microbial communities on mushroom development, and that's all made possible from our initial funding from HITS. From that, I'd like to acknowledge the Devome team. I'd like to acknowledge everyone that works at the Mushroom Research Center for helping us with our crops. Dr. Tatiana Larimore of the Proteomics Core for making my work possible. And obviously a big thanks to the Hook Innovative and Transformational Seed Fund and the Microbiome Center for supplying me with funding towards travel, which helped in my training for Metaproteomics and for inviting me to speak today. And thank you all for listening.
Talk. Let's stick with the tradition. Any questions from trainees?
I had a question about passaging, and is it a common practice in like mushroom systems amongst producers, or is this like a new sort of practice that might be integrated into mushroom systems?
It's not a common practice in the mushroom world, but you can make analogy. It would be like fecal transplant or something similar, you know, like for animals or plant system. Up to my knowledge, that's the first time I think somebody tried that approach of transferring from a tool or different organisms, communities to a different stage of life, so on. It's not common in the commercial world, either in R&D. For mushroom purpose research, yeah.
I would consider it slightly akin to the idea of, like, say, suppressive soils with disease, where you are choosing a substrate at a point of interest and trying to reintroduce it and see if you get a beneficial phenotype from that. So that as well, slightly not the same, but kind of same type of concept.
I have a question. I'm sorry if I didn't. I lost. When did you add antibiotics to compost? At which stage of compost preparation?
The inoculation day.
Inoculation day.
We made the phase. Are you familiar with composting?
Yes.
For mushroom? Phase 1, phase 2?
Yes.
Phase 2 compost is ready to be inoculated. that moment. When the compost was ready, finished the composting, we spread, we mixed the solution.
Yes, after pasteurization, you add antibiotics.
Right. And encasing was at the casing day, the day you prepared the peat moss material.
Thank you.
Hi, I was really struck by the high diversity in the composts, that all those alpha diversity plots seemed like a ton of different types of bacteria. So I'm curious if that creates a limitation for work like metaproteomics, where it's going to be really database restricted. And if you were able to annotate a large fraction of those bacteria, or if we're kind of talking about the function of potentially a very small fraction of the total bacteria.
So you're asking, would you still see functional annotation based off what could be a very small? Yeah, it's a bit of a black box in a sense, and I mean it's the old trope of the better genomes we have, the more the better our annotations would be. So there is always a level to which that functional annotation will suffer from the lack of fully resolved genomes, things like that as well. We can still capture that something is there. We just might not capture what it's doing. So even with some of my data sets, we will still see hypothetical proteins, unknown functions, and things like that. So they're still captured, but yeah, you're right. Particularly when it comes to pathways and things like that, you're always looking at a more specific lens on the wider subject. It is very difficult, and that's why with metaproteomics we do like to use core plots and things like that, because it really spells everything out as much as possible. But yeah, you are kind of trying to find a needle in a haystack. Yeah, in that sense.
I want to piggyback back off of that because that's where my question was headed to. First of all, congrats on the ton of work you guys have presented and the foundation it builds from here. So when you match the metaproteome to the metagenome, I guess a key question for you or the field would be what percent of the metagenome is represented by the metaproteome, barring that read depth matters in both cases. But is there conversation about sort of what is expected? And I'm guessing it's low.
To a degree, yes. So there is initiatives like CAMPI, which is we're trying to consolidate metaproteomics research and have defined, I suppose, steps as to how we even extract our proteins down to how they're analyzed. So there is kind of a consensus that's being formed as years go on. Within that, within soil microbiomes in particular, it's a huge challenge. 5% could be sometimes the amount of, if your full PSM field is present, and that's so many PSMs, 5% of that might actually map to what you got from the mass spec. If it's the human gut microbiome, 50%. Anything human related, model organism related is going to have a much bigger resolution. But it still stands to the point of how difficult that sample is. And even within soil is hugely diverse. Like casing is full of humic acids, composts have different interfering substances. So even just saying soil is such a blanket term for all these diverse, you could have biocrusts and things like that. But generally I've seen sometimes as little as 5% is being mapped. But what I will say to that is you're still probably going to capture the big hits within that 5%, and maybe the really small numbers might be filtered out. But if they're so small, we have to beg the question of, if the biomass isn't there, is it that important? But that's a question for the field. Yeah.
Thank you.
Let's thank you, our speakers. Yeah.